in Madrid, and her line of work is sensory biology in mosquitoes. She did her PhD at the Universidad Autónoma of Madrid, and then she did several postdocs in Germany, and then at UCL, where she became an early career researcher leader group. She has been working on sensory biology from different perspectives, from the cellular level of antenna, but also to the behavioral level that links her research with our research, which involves more ecology and evolution. So I hope you all enjoy her talk, as I did when I met her first in Greece several years ago. Thank you so much.
Thank you. Okay, thank you very much for the invitation and for coming. So, yeah, I don't think this needs much introduction in this audience, but as you know, mosquitoes have been, but are becoming perhaps an increasing public health challenge in Europe. So this map just shows you, as an example, the expansion of the tiger mosquito, Aedes albopictus. You can see in less than a decade how it has been spreading across Europe, and this is probably, you know much more of this than me, but this is a species that has been traditionally found in Europe and in Spain, and that is becoming or it's a vector of emergent diseases. So altogether, this picture poses, let's say, a public health challenge, and we need to develop novel control tools to face this public health problem and to control mosquito species. As you might know, so mosquitoes act as vectors of different diseases, and they cause approximately 750,000 deaths a year globally, and these are some of the important vectors and are the ones that I'm gonna somehow talk about in the talk. So Culex pipiens, that it's very well known by you, which acts as vector of West Nile virus, then Aedes albopictus, the tiger mosquito that I was mentioning before, that it's spreading very, very quickly across Europe and in particular in Spain, and act as vector of different arboviruses. And then Anopheles gambi, which is one of the main malaria vectors that I'm gonna talk most about in my talk, because it's the species that we have been studying in more detail, that it's found in sub-Saharan Africa. And because Anopheles spreads malaria, we've been basing our research on the tools that are used to control malaria mosquitoes. And as you might well know, malaria mosquitoes are mostly controlled using two tools, which are insecticide-treated bed nets and indoor residual spraying. And they have been very successful in controlling mosquitoes and in preventing disease, but they have some challenges. And it's first of all that they are targeting just a specific stage of the life cycle of the mosquito. So they are targeting females that are either host-seeking or just that have blood fed, meaning that there are other opportunities missed that could be exploited to control mosquitoes. Also, it leaves mosquitoes, for example, that are biting outdoors, or species that are adapting behaviorally to avoid these control tools. They are untargeted, and therefore there's residual malaria transmission still taking place. So what we propose is to contribute with alternative tools, and one of the obvious life stages that could be targeted for this is the mating behavior. If we are able to stop or to disrupt or to reduce mosquito mating, mosquito reproduction, we would obviously have a great impact in mosquito populations and therefore in disease transmission. And how do mosquitoes mate? Well, mosquitoes mate in swarms, which are mostly in swarms. I'm not going to say only in swarms, but mostly in swarms, which are aggregations. It can be of up to thousands of mosquitoes that, again, for Anopheles gambi, for malaria mosquitoes, take place at sunset. They are mostly composed of males, with just very few males entering the swarm to mate. And how this recognition, this mating pattern recognition takes place is acoustically. So mostly the male will detect the flight tones of the female and will chase her to mate. So from a very simplified perspective, we can say that, okay, the male and the female flight tones are clearly distinct, so the frequencies are very different. And we can say, and this is very general, but we could say that there are differences also across species, and therefore it's like the signature that they use to recognize the mating partner. So what we are trying to understand, first of all, so our research is also quite fundamental, so we try to understand how this process takes place, and then we try to see if we can develop pharmacological or even genetic disruption strategies. And because the way this recognition is acoustic, we are very much interested in the mosquito auditory system, the mosquito ear. And the mosquito ear is it's an antennal hearing organ. You can just see it by looking at the mosquitoes. And it's composed of two main parts. So the flagellum would be the sound receiver, so our eardrum, that will vibrate when there's a sound in the close vicinity of the mosquito. So in principle, the mosquito auditory organ is specialized in short range hearing. So they shouldn't be able to hear sounds that are far away, but sounds that are emitted nearby. So when the mosquitoes are flying around, the sound emissions will deflect the flagellum, and this deflection will be ultimately transmitted to its base, where the auditory organ is located, which is called the Johnston's organ, after the researcher that discovered it. And actually this organ is very complex. So just to give you an idea, in males, because the great difference between males and females, but in males the organ harbors around 16,000 auditory neurons, which is more or less the number of hair cells in our cochlea. So it's a very complex organ, and it's, as I said, the sound receiver, the flagellum, has some cuticular projections emerging from its base. When the flagellum vibrates, these projections that are called prongs vibrate, and ultimately this vibration will be transmitted to the auditory neurons, to these cilia, which are the sites of mechanotransduction. And these neurons are organized in units that contain 2 to 3 neurons with some support cells, and these units are called scolopedia. And think of this is just like a cut through the organ, but actually it's like an upturned umbrella. So we have 70 of these cuticular projection emerging from its base, and then it will add to these 16,000 neurons. And this is just a picture, like a immunostaining, but you can see a red neural marker, a green cytoskeletal marker, but you can see how this looks. So the flagellum will be here, the prong emerging from the base, and then the auditory neurons with the cell bodies located in the periphery, and the cilia pointing towards the prongs and connecting to them. And then there's this massive nerve, the auditory nerve, the antennal nerve, that contains the axons from all these neurons, and that travels to the brain carrying the auditory information. And some of the main characteristics of the mosquito auditory system, what are they? Overall, in most species, this organ is highly sexually dimorphic. So this is the case of Anopheles gambi. Red, you can see the neurons, and you can see that even in females, we are a bit zoomed in, so it's even smaller, but you can see how different the organ is in males and females. Some species, for example, Culex pipiens, I'm not sure if anyone has looked at that, but I think the difference is a bit smaller, but still it's clearly distinct. And then one feature of the mosquito auditory system that we described a few years ago is that it's not so. There's not only an auditory flow of information from the Johnston's organ to the brain, but actually there's a flow back. We can say bidirectional information. So the brain controls how the auditory function takes place. So the brain sends some neurons, which are called efferent neurons, back to the Johnston's organ that will release different neuromodulators, different neurotransmitters to control the auditory function. And this is very unique because although in vertebrate ears, these efferent systems are found across all vertebrate ears, in insects, mosquitoes are so far the only insect where this has been described. I'm not saying it's the only one that has it because I'm pretty sure it's not, but it's the only system where this has been observed. So it's very unique. And what we did is we tried to characterize which neurotransmitters were released by this efferent system, by this efferent network, and we saw a complex neuromodulatory network. This is all immunostaining, so we it was targeted. We know what we were looking for. We don't know what else might there be, but you can see that we found octopamine being released there, serotonin released there. So these are biogenic amines that are very well characterized as being neuromodulators of mosquito behavior. And then you can see GABAergic innervation, a massive GABAergic innervation of the antennal nerve, which is happening in different locations within the organ, suggesting different function, and at the end of the day, suggests great auditory plasticity. And along the talk, I'm going to focus mostly on octopamine and GABA, what we know of what they are doing. We also look at the efferent system across species. We look at these three, Aedes aegypti, Culex cinquefasciatus, and Anopheles gambi. We found, I mean, these are males and females. The efferent network is present in all species. It's quite well conserved, so the pattern is quite similar. And I want to remind you, these were species that diverged very long ago, more than 200 million years ago, suggesting that this efferent system is an intrinsic component of the auditory system of this family that they require for their auditory behaviors. And as you can see here, Anopheles gambi is the case where the sexual dimorphism is more extreme, but you can see that these organs of Culex cinquefasciatus, Aedes aegypti in females are a bit more complex than Anopheles gambi, and they present some efferent fibers. And this is all hypothetically. What we think is that actually this efferent system, the difference that we found across species, might be related to the sensory ecology in the swarm. So at the end of the day, what the mosquitoes use their ears for is to detect the mating partners. And as we were discussing, the mating partner detection takes place mostly in the swarm. So you can, Aedes aegypti, they form very small swarms close to the host. Culex cinquefasciatus, Culex pipiens, we don't know, but for middle swarms, while Anopheles gambi, there's an extreme, so very big swarms, they will be very noisy inside. So we obviously think that this sensory ecology is influencing how the efferent system looks like. Although how this is being shaped, we are still trying to understand. So what we do to study the mosquito auditory system is we use this methodology. So we will mount the mosquito, we glue everything except for the joint between the flagellum and the Johnston's organ. So we want to see how the flagellum is vibrating. It's the only movement that we want in the mosquito. We direct a laser to the tip of the flagellum so we can record the mechanical vibrations. We inject an electrode in the antennal nerve so that we can record electrical signals. So how is the organ responding to sound or to the mechanical stimulation? And we mostly use electrostatic actuation to deflect the flagellum. So we don't use sound, we use this mechanical stimulation because we can control better the type of stimulation that we are providing. But at the end, it's just a mechanical stimulus. And then we will inject different compounds depending on what we want to study. And very briefly, because I don't want to get into much in detail, but just very general characteristics of the organ. So the organ presents frequency selectivity. It means the organ is tuned to specific frequencies that are biologically relevant, which will be the wind beat frequencies of the conspecific partners in a simplified way. And then there's an active sound amplification. So they can actively amplify the sound. So if we present quiet sounds, they will be able to amplify their compare to louder sounds, and this has some implications in terms of the auditory processing on the auditory function. Another characteristic of the mosquitoes is that they present spontaneous activity, much as our ears. So it means in the absence of any stimulation, the flagellum is vibrating, and it's vibrating with a specific frequency that will act as a filter. So they will filter the auditory information that they are getting from the environment. So it's relevant, the type of spontaneous activity that they present. And while females present these spontaneous vibrations of around 200 hertz, males, we find two different states. Some males that we call quiescent are presenting these spontaneous vibrations around 500 hertz. And then we have these other males which are called self-sustained, well, that present these self-sustained oscillations that present a spontaneous activity of around 350 hertz. But I want you to point you also at something. If you look at this axis, the amplitude of these vibrations is like 1,000 times stronger than in quiescent states, meaning that these vibrations consume an energy, and that they might, in principle, have a biological meaning that I'm gonna try to explain you later, what we think these self-sustained oscillations are doing. Yeah, sorry, so those are the frequencies of each and the amplitudes of the vibrations. Another characteristic of a mosquito auditory system is that the mechanical and the electrical tuning in males do not match. That means that if you present this, this would be our stimulus from, in this case, from 0 to 600 hertz. The amplitude of the responses of the flagellum and of the nerve, or the amplitude of the strongest responses for the flagellum and the nerve, happen at different frequencies in males. So you can see this one, the dashed line would be the mechanical tuning, so how the flagellum is responding. The continuous line is the nerve response, and you can see that in females these two mostly match, but you can see that in males there's a clear mismatch. So while the flagellum is responding to frequencies of around 320, 350 hertz, the nerve is mostly responding to frequencies around 200 hertz. And this is quite non-intuitive. So there was a question, why is this happening? What's happening here? And I'll go back to this graph here and this is now quite well established. So basically, what has been proved that is taking place is that, again, here you can see electrical tuning. So the nerve response tune around 200 hertz and the mechanical tuning around 300, 400 hertz. And you can see that actually the mechanical tuning is coinciding with the female wind beat frequency. So the female wind beat frequency is around this 350, 400 hertz, while the male wind beat frequency is higher. It would be 640 hertz, something around that. So what we think is happening is that these two tones together, and this is all biophysically, it's a biophysical property of the system, will generate some distortion products. So mixing these two tones in the mosquito auditory system, we exactly don't know where, probably in the flagellum, but generates these low frequency distortion products that around 200, so it's the difference of these two tones is male minus twice the female wind beat frequency, and as I said, this is all biophysical properties of the system, but these low frequency distortion products is actually what the nerve is picking up. And if you think about this is very smart, because basically what is happening is that the loudest tone that a male mosquito will hear will be always their own flight tones, because they have the wings on their backs. They cannot avoid that. So they actually use their own tones to hear the female. They are transforming the sound emissions of their own tones into something that is allowing them to detect the female. So a very smart strategy. The second point is that in principle mosquitoes need to be flying to hear the female. If they are not flying, they will not be able to generate distortion products and they will not hear. And what happened? How are these self-sustained oscillations then adding to this system? Well, these self-sustained oscillations, again, the numbers are moving a bit around because technically the frequencies will change depending on the temperature, the experimental setup, and so on. But basically, what we think is happening is that the male self-sustained oscillation that you can see is very similar frequency to the female wind beat frequency, amplifies the female wind beat frequency. So by generating this tone already in the flagellum, the amplitude of the distortion products is stronger, and therefore the females will be more audible. The male will be able to better detect the female. And again, this is just very briefly, we don't know, we don't know what's causing self-sustained oscillations. We know that in the lab sometimes they happen, sometimes they don't happen, but we don't know why. We don't even know if in a swarm they are showing self-sustained oscillations. That's our prediction, but we have not been able to test it. But what we know is that if we block the efferent system, so this efferent network that was innervating the Johnston's organ, if we block it using tetanotoxin or tetanus toxin, the self-sustained oscillations automatically start. So somehow the efferent system should be able to induce them. Okay. There has been a bit about the basics of mosquito here. Now I will move a bit more towards what we do in the lab. Because the mating behavior and swarming behavior is such a rhythmic behavior, we are very much interested in having this perspective, this, let's say, rhythmicity perspective in the lab, let's say. So we are very careful in maintaining the circadian entrainment of our mosquitoes. And I just want to show you here, like, this is just how even, obviously in nature, the behavior of mosquitoes is rhythmic. Also in the lab, we can mimic this. So if we entrain them in the lab, if we keep them in incubator, and we keep them in one of the chambers that you were using, or in this case, we use these locomotor activity monitors, we can clearly see how Anopheles gambiae is very quiet during the day, becomes active during the night, and present this very clear peak of activity, which we will call swarm time, because transition from light to dark is when swarming behavior happens. While, for example, Aedes aegypti, which is a more diurnal mosquito, we can also clearly see this, how they are active during the day. So basically, and we can see a reflection of this in the auditory system as well. So, and again, very briefly, but we see some changes in the mechanical tuning of males during the day. So the frequency of the auditory tuning, so the frequency that they are mostly interested in, increases a bit during swarm time, so there should be a modulation of that. We also know that there is an increase in the flight tones, so it goes together, because obviously the system needs to work together. Flight tones and auditory tuning goes together. We also see an increase of the flight tones in Anopheles gambiae at swarm time, going together, and I will talk later about something that relates to this. We also know that the anatomy of the system changes. I don't think this happens for cool excipients, but in Anopheles gambiae, the fibrille of the flagellum that males have will only become expanded at swarm time. So they have it closed during the day, and it will be just expanded at swarm time. And then else, I don't know if this video will work. Let me see. Oh no, sorry. But basically, mosquitoes, male mosquitoes are attracted to sounds. That's the phonotactic behavior, how they will chase the female. And that in Anopheles gambiae only happens during swarm time. So there's also a modulation of the auditory behavior itself during some time that we still don't know how it's happening, because from a, let's say, electrophysiological perspective, they hear all day, but from a behavioral perspective, they only respond to sound during swarm time. We still don't know why. Okay, so what we did in the lab, we first tried to do some transcriptomic analysis to understand how this is happening, what's the different system doing here, what type of modulation do we see along the day. So we collected mosquitoes every 4 hours, we conduct RNA sequencing, we separated the Johnston's organ without the flagellum, just the Johnston's organ and heads, and then we selected using some gene ontology term analysis. We selected mostly neurotransmitter receptors, neuromodulator receptors, G protein coupled receptors, and then we observed that actually, and these are a long list, but that there's a lot of neurotransmitter and neuromodulator and G protein coupled receptors expressed in the Johnston's organ. Not all of them, you obviously need to be target of the different system, because there can be some release of hormones or release of neurotransmitters in the hemolymph that will reach the organ. But we saw, if you remember, I was talking about octopamine, serotonin, and GABA receptors, and they are, I mean, we saw it, the release of neurotransmitters, and they are expressed there. This is just a slide to bring this all together. So where these neurotransmitters are released and the type of receptors that we observed expressed in the Johnston's organ. And in particular, we were interested in this one. 2886 is an octopamine beta-2 adrenergic receptor. Why we were interested in this? Well, it's the most highly expressed octopamine receptor, and obviously the octopamine pattern is very beautiful, so we wanted to understand what octopamine is doing. And we were quite lucky, I have to say. So what we did, we generate knockout mosquitoes. We do this by inserting a GFP in the open reading frame of the gene. And we expect that introducing this, which is 3 kilobases, will disrupt the gene function. This is how it's being inserted. And then we analyze these mosquitoes. And I said we were lucky because we observed a very nice first phenotype. And basically is that these mutants do not expand the fibrille at swarm time. So basically they will remain collapsed along the whole day. We already knew that this is what octopamine does. So if you inject octopamine in an Anopheles gambiae male at any time of the day, they basically will expand the fibrille. So it's induced by octopamine and it's mediated by this receptor. So if we inject octopamine in wild-type mosquitoes, we see fibril expansion. This was the baseline. So the baseline at swarm time no mosquitoes are expanding fibrilla for the wild type. Sorry, all mosquitoes have erected fibrilla for the wild type. No one has for the mutant. Then we mount them, because this is important. When you mount them, they all collapse it. So it's like a starting state. And then we inject it, and you inject octopamine. You can see most of the wild type mosquitoes erect the fibrillae, and none of the mutant do. So it's already proven an important role in the mosquito auditory system. We then analyze further auditory parameters, and again we are analyzing this the mechanical and electrical tuning. So the frequencies, the frequency that the flagellum is most responsive to, and the frequency that the nerve is most responsive to. And you can see that. So in blue would be our wild-type mosquito. If we inject octopamine, we see a very clear increase both of the mechanical tuning frequency and of the electrical tuning frequency. But if we do this in the mutants, this doesn't happen. So they don't change they are not responding to octopamine. Again, proving that this receptor is very important for octopamine auditory role. And we checked, and this is actually reflected in mating behavior. So these are experiments in small cages. And you can see that if we cross different combinations of male and female wild type and mutant mosquitoes, when the males are mutant, there's a very severe decrease in the mating rate, in the insemination rates. So we will perform the cross, let them mate for 2 or 3 days. I think this is 3 days. Then we will dissect the spermathecae. We look for the presence of sperm. If there's sperm, they are mated. If there's not sperm, they are not mating. And this is what we show here. So in this process, how many females were inseminated. And you can see that when males are mutant, doesn't matter the female genotype, there's a very clear effect on the mating rates. And again, I said we were lucky because this is obviously already a very nice phenotype, but then when we look at the females, they don't present mating defects, but they are actually sterile. So you can see in this, they are the same crosses, but then we look at what's happening with the eggs. And we saw that when females are mutant, there is no larvae. So these eggs are not able to hatch. So basically, if I go back to our initial plot, if we are able to disrupt this receptor, we would be able to disrupt mosquito mating, but also perhaps it could also be used for indoor residual spraying because females will not be able to develop their eggs appropriately. So we wanted to follow up a bit on this, and there's actually one insecticide, which is amitrad, that is already, it's an agonist of this receptor, and it's used for mite control. So it's used as a pesticide. And we try to understand whether it can be applied for mosquito control as mating disruptor. And this is just to prove that actually if we expose the mosquitoes to amitraz, you can see that they expand the fibrillae, so it's acting as an agonist of this receptor. And if we do this in our mutant mosquitoes, there's no fibrillae expansion, meaning that the in vivo target, at least one in vivo target of amitraz is our receptor. But amitraz exposure doesn't cause mortality in mosquitoes. That's why it's not used for mosquito control. So what we did, and this is just now being reviewed, so this will be updated very soon. But the whole message is the same. But we expose mosquitoes to amitraz in small cages, large cages, and in the semi-field. And we actually saw a small, subtle decrease in mating rates in the laboratories, both in small cages and in large cages, but we didn't see any effect in the semi-field, meaning that perhaps in the nature they are able to overcome the potential disruption caused by amitraz. We don't know, but clearly it doesn't seem to be a good mating disruptor. And we didn't see any effect on female fertility. So what we try to do is to see if in any way we can suggest that amitraz can be modified to increase its potency on the mosquito receptor. And I was just telling you, so amitraz is used as a pesticide, and it's used to protect bees from these mites that are parasitic mites. And amitraz kills these mites. And so so we, and this was also reported by someone before, but we express the mosquito, bee, and mite receptor, and then we look, and you can see that actually the mite is more, so Amitas has a higher affinity on the mite receptor, that's why it's killing them. While if we check for octopamine, it's almost the other way around, so it's a bit maybe more potent on the mosquito receptor, but we try to understand this a bit more. So we look at the binding bucket and the amino acid, different residues, and then by literature and by conservation across species and so on, we selected 3 residues and we mutated them in different combinations. So perhaps we just took the mosquito receptor and mutated it into a bee receptor, or mutated it to a mite receptor, and then we saw changes in the effects of amitraz on these mutant receptors. And this is just without further detail, but we are trying to understand if we can modify, well, in this case we are modifying the receptor, right? But trying to understand what's the important parts of the amitraz structure that could be mutated to, okay, could be modified to increase the potency and perhaps potentially to disrupt mosquito reproduction. And this is kind of the end of the first part of the talk. So I just want to, so the take-home message, what would it be? So the octopamine beta-2 adrenergic receptor, we believe is a good candidate to develop reproductive control strategies, but amitraz is not valid, but could potentially be modified to increase its potency against the mosquito receptor. So we think it's a good starting point to generate new molecules. Okay, and I'm going to change quite a bit because I'm going to talk about GABA. If you remember, GABA was another of the efferent neurotransmitters that was mostly released in the antennal nerve. So this antibody is actually not recognizing GABA, it's detecting an enzyme that is required to synthesize GABA. So, we assume it's showing a GABAergic innervation. And again, in the transcriptomic analysis that I was talking about before, we just saw different GABA receptors being expressed, and we were particularly interested in this one. First of all, because it was the one most highly expressed, RDL, resistance to the eldrin, and second, because it's a target of insecticides. It's a GABA-gated chloride channel, so it means when GABA binds to it, it opens and lets chloride flow into the cell, so it's hyperpolarizing the neuron, it's reducing, in principle, its responsing. GABA is an inhibitory neurotransmitter. And then we saw that it's mostly expressed by these distal neurons. So this is in situ hybridizations, and we saw that there's differential expression across the organ and potentially more highly expressed on these distal neurons of the Johnston's organ. We analyzed the auditory effects of RDL, and the way we approach it, we couldn't generate knockouts because it would be lethal. So we used picrotoxin, which is a very well characterized antagonist of RDL. So basically we are blocking the receptor. And what we saw, and again this had already been reported by in Anopheles Stephensii, is that if we apply picrotoxin to the mosquito, the fibrillae become expanded. So it's pretty much doing what octopamine was doing. And very interestingly, if we apply picrotoxin to the octopamine beta-2 adrenergic receptor mutants, this doesn't happen, meaning that GABA should be acting upstream of, in this case, octopamine or the octopamine receptor. So GABA is inhibiting, we block the inhibitor using picrotoxin, and then octopamine is released and fibrillar erection is taking place. So we are inhibiting the inhibitor, and therefore we see the effect. And again, this is just showing the same that I was discussing before. So we see if we inject picrotoxin, if you remember when we were injecting octopamine, we saw an increase in the frequency of mechanical tuning. So we see the same when we inject picrotoxin, but if we do this in the mutant, we don't see this effect. So our conclusion from this is that the mechanical effects of RDL are actually mediated through octopamine. But then we also look at the effects. So actually, when you inject picrotoxin to a mosquito, the very clear, it's been, okay. the very clear effects that you see is that the nerve starts spontaneously, so there's no stimulation here, it fires a lot. So, so you can see this is the spontaneous activity that we see in the nerve before we inject picrotoxin. These are not spikes because we are recording from its compound action potentials. So we record from different neurons, but at least are peaks of compound action potentials that we observe. You can see that if we inject picrotoxin, this becomes much stronger, and we still see this in the mutant. So we assume that this electrical this electrical effect of picrotoxin is independent of octopamine, and this makes sense because if you remember when I was showing you where GABA is located, it's basically innervating fully the organ. So this should be a direct effect of GABA on the auditory nerve, while again, and in this paper we show the model and everything, but while the mechanical effects are probably mediated outside the central nervous, well, in the thoracic ganglion through some octopaminergic efferent neurons. I mean, it's a bit complicated, but basically the important thing here is that it seems that RDL might be eventually modulating the auditory sensitivity, because if it's able to, when we are blocking RDL, we see a very strong increase of the spontaneous activity in the nerve. It means that normally RDL is somehow inhibiting that the electrical activity. So basically from this we propose that it should be able to control the auditory sensitivity. And from here, I think this is actually the part that is going to be most interesting to most of you. I just told you that so RDL was first described as the locus where target site mutations to the insecticide dieldrin are found. So dieldrin is a organochlorine insecticide that was very widely used for mosquito control in the 70s, and then it stopped because of toxicity and resistance issues. So there was very widespread resistance, and still, so since the 70s, it's no longer used, but still these resistant mutations are very widely found in mosquito populations without a clear understanding of why. So there's no more selective pressure because the insecticide is no longer there. Why are these mutations still there? With some caveats that we can discuss later. And basically these target site mutations are two. They are all located in the codon 296, and these are these alanine to serine and alanine to glycine resistant mutations, and this is just a map that shows that these mutations are still very widely found across mosquito populations in sub-Saharan Africa. So we thought, okay, it's interesting that RDL is modulating the auditory sensitivity. These mutations are kept in nature without we really understanding why. Maybe there's a connection between the two. So what we did is we analyzed some insecticide resistant line that we had in the lab, and this is an homozygous resistance. So this carries this so serine homozygous residues in the position 296. By opposite, you can see this is the wild type, alanine. And we perform these phonotaxis assays where basically we play a sound that mimics, well, this sound, you can see we play sounds, pure tones of different frequencies, but you can see that here around the frequencies of female flight tones is where male mosquitoes are more responsive to. But you can see that actually mutant mosquitoes, so resistant mosquitoes, are more responsive to those sounds compared to wild type, meaning that they are more attracted to sound, meaning that maybe they are able to better recognize the female. And we conducted also some additional mating experiments. So these were experiments that were conducted in a low noise, because we were calling this silent, and they told us you cannot call this silent. Low noise and high noise incubators. Unfortunately, our incubators are already very loud, so we cannot do silent conditions. So the low noise is 70 decibels as opposed to the high noise, which I think is 93 decibels. And then we compare, so we cross, these are female resistant mosquitoes and female susceptible wild-type mosquitoes, and we cross them with resistant and susceptible males, and you can see that we find an interaction between the male genotype and the mating rate in the different incubators. So basically there's an interaction between the male genotype and the noise levels in our independent variable that would be the mating rates. So because of the model, it's difficult to say the direction, but together what we are proposing here is that in the lab we have evidence that these insecticide-resistant mutants are first of all more responsive to sound, and then that this genotype can influence the effects of the noise levels in the mating rates. So yeah, so based on this, what we went to the field and we tried to see if, so basically our hypothesis would be, okay, this advantage, this auditory advantage of the insecticide-resistant mosquitoes is allowing or is keeping or pushing the maintenance of these mutations in nature. So what we did collections in Central African Republic, and we tried to collect mosquitoes from urban and rural areas, being our assumption urban areas will be noisier than rural areas. We might establish this association between urban areas being noisier and so on. So this is what we did. We collected mosquitoes and we collected noise levels as well, environmental noise levels. And you can see that we indeed found increased frequencies of resistant mosquitoes in the city of Bangui compared to rural areas. And you can see that this was stronger in Anopheles colutzii mosquitoes. So these are members of the Anopheles gambi complex. And this is even stronger for these A2S substitution, so A2S mutations. I have to say, and this is a limitation of our study here, we found heterozygous. So most mosquitoes we found were heterozygous, not homozygous as in the lab, but there was a very clear enrichment of resistant mutations in the city. We also, independently of whether it was an urban or a rural location, we looked for an association between the noise level and the RDL genotype, and we found an association between higher noise levels with resistant mutations, independently of whether it was an urban or a rural location. And when we look within the city, because we really wanted to prove that this independently of a rural-urban area, because there are many confounders that can take place there. And we saw that within the city, in the two highest and the two quietest locations, there is again an enrichment of resistant mutations in the highest areas compared to the lowest areas, and this is statistically significant. I didn't write the significant levels, but even within the city, we find this higher proportion or this enrichment of resistant genotypes in the noisiest locations. And we also looked at the mating, at mating in the field, and this is difficult to do. We couldn't do collections from swarms. So what we look at the female insemination rates that were captured from the field, and this is, and you can see here, so the three genotypes: susceptible females, resistant females for A to G, and resistant females from A to S. And you can see that in rural areas, susceptible females present higher mating rates compared to the urban areas. So you can see here mated in the rural compared to mated in the urban areas, and you can see that there is a decrease in the mating rates of the susceptible females in the city, suggesting that there might be some issue that females might find it more difficult to mate in a city compared to a rural area. We connect this to noise, but of course there could be caveats around that. And then again, we find this association between the noise levels and the insemination rates in the susceptible females, which is a negative association. So females in the susceptible females in the noisiest areas are less mated than in the quietest areas. And altogether, we think that this is a very interesting and new hypothesis, and it's basically that So resistant mutations might not only be detriment, I mean, they might not only provide as an advantage towards surviving to the insecticide, but perhaps they are also causing other advantages that can be eventually selected, and that can, if our hypothesis is true, that is what we are trying to prove. Basically, it would mean that these mosquitoes are better at mating, and that they might be better to colonize cities because they are able to detect the females better in the city compared to the urban area. So basically we think this angle is very important, because when applying, for example, insecticides, insecticides are mostly targeting neurotransmitter receptors, and no one is looking at these potential side effects of insecticide use that might be altering mosquito behavior, and that might be even selecting fitter mosquitoes under certain conditions. So, okay, so, and with this, I just want to mention, we are part of this staff exchange, which was what was we were organizing the meeting earlier. So this is where we are trying to develop novel tools to disrupt mosquito mating behavior, and there are different partners here that we are working together to try to develop these tools. Well, not develop, we are still it's very fundamental still, but it's our end goal, trying to see if we can develop mating disruptors or even apply our knowledge to genetic control strategies. Yeah, this is, so most work has been done with this team, because I was before in University College London in the UK, and this is the new team. We are CISA, Centro de Investigación de Sanidad Animal in Madrid, and we are starting, well, it's been a year, and we are starting to set up everything here. So I would like to thank them and thank you for your attention. I'm happy to take some questions if you have any.
Okay, super good talk. Yeah, thank you. And do we have any questions in the? Questions in the room?
Go ahead and start. Hi, thank you, Marta. It was a really nice talk. So I was wondering if, I mean, I know that you already studied it regarding the relationship between resistance and the difference in the urban areas and rural areas. And I was wondering if you know the history of the Eldrin use, if it might have been higher in urban areas than in rural areas. I don't know exactly how the Eldrin is used, so I was just wondering about that. Okay, so yeah, I didn't mention it during the talk, but that's, so obviously, like what they think, that the main theory is that there's cross-resistant mechanisms to other insecticides. So there are other insecticides that are being used, like fipronil and other organochlorines. There might be some cross-resistant mechanisms, but it's not clearly established. So these mosquitoes are resistant as well in the lab to fipronil, but there's not a clear association. Let's say there's not a as far as I know, no one has shown that fipronil, because fipronil is also a pesticide, is more highly used in urban areas compared to rural areas. So, I mean, but obviously that's playing a role, because RDL-resistant mutations are also found in other insects. So it shouldn't be just related to mating behavior in mosquitoes. But what we are proposing is that this might play a role, and that this might play a role, for example, as well in assortative mating, so that this might be contributing to the maintenance of these mutations in the field, and that, again, that it could be playing a role as well, specifically in urban areas, and that it might facilitate as well mosquito colonization. So if the mosquitoes are able to better detect the female, that still no one has really known, again, as far as I'm aware, how environmental noise is affecting mosquito mating behavior. That needs to be characterized. As I said, mosquitoes are specialized in very short range hearing, so in principle, a sound that is far away shouldn't affect them. But if the sound is very loud, that can affect as well how mosquitoes are swarming, but that has not been studied. So what we are proposing of this study is still a preliminary. We think it's a very, because we provide evidence to show that, but obviously this needs to be further investigated. There are different things that should be done to really understand how that's happening, what mechanism is that. For example, why do we see females in principle are not the ones detecting the male? So, but what we are showing there is that there is an effect
of noise on the mating behavior of females. So why is that happening? We think that maybe there's, as I said, assortative mating mechanism, so that's why if males are able to less detect the female in the city, they might prefer their RDL mutants, and that's why, you know, that, yeah, so it's complicated, yeah.
Well, thank you for this impressive talk. Congratulations. So I guess that the goal is not to eradicate mosquito populations, because that's impossible, but to keep mating rates to a given level. So what's the estimate of the right population size and mosquito populations to keep to assess them as acceptable, and that they will not create problems? So when you decide that the actions taken, you know, will be successful.
So you mean in general, not only for mating disruption, but rather for overall? I think maybe they can even reply to that better than, so I mean, I think that the end goal would be to eradicate a specific species that will, but as you say, that's impossible, so maybe the goal is more to eliminate, but I think that's a very complex question, because you would need to see how disease transmission is affected by the mosquito population numbers. I really don't have an answer. I don't know how low you need to bring the population for that. Probably that's the dependence on the disease, on the pathogen that is being transmitted, on the setting. So I really, like what we would like to do is, like what we would like to contribute is, our ideal scenario is if we could generate a molecule that could really decrease mosquito mating rates. And again, this is not going to be substitute any of the other tools. That obviously the females are the one biting, so you want to stop them from biting. That's what the bed nets are doing, apart from killing them. But what we are trying to say is, like, okay, modifying mosquito behavior is not used at all as a tool. We should also explore that, and in particularly, and that's not evaluated, so they will not, there are no protocols to evaluate. So I think the end goal of the tools needs to be killing, but it's also like, okay, maybe the end goal of the tool, if you are having an effect on mating, if you are having an effect on other behaviors, could also contribute to mosquito control, and it could expand a lot the range of molecular targets that you could use. So that's a bit what it's also kind of providing a proof of concept of applying this, but yeah.
I have another one, if I may. So I would say that any use of chemical will start an arms race, I would say, right? So in your new project, what you are trying to evaluate is other mechanisms, like changes in behavior, complicating the way that males detect females or something like that.
So you are talking, for example, about sound traps or things like this, or
something like that.
So sound traps, I mean, we don't that would be perfect, because that would be ecologically friendly, you are not releasing any chemical, but it's not easy. So sound traps have been tried to be developed along years, decades, and they are used, I think, for surveillance purposes against Aedes. They are incorporated to some traps that they would be chemical traps, but then if you incorporate a sound, then you increase the capture of males, for example. And they are being explored. The only trick, the only difficult challenge is that, as I said, in principle, mosquitoes are specialized in short-range hearing, so they need to be close to the sound source. If you want them to be attractive from farther, you need to play a louder sound, and they will be able to hear them, but for the acoustic energy to reach them, then you need to increase the emission. And then that's, it's difficult to have a very loud sound, because it's not. But there are labs that are exploring that, and there might be ways to optimize the sounds that are being emitted, because I think perhaps we are not using the right sounds, the right type of stimulation, and there are labs that are doing that. So perhaps they might be able to find a better stimulus that then can be played and attracts more males, but that would be for males, but it wouldn't affect the female, it would affect the males, which are the ones responding to the sound.
Hi, thank you very much for the talk. It's a little bit related to the previous questions. So you said in the beginning that swarm mating is the main mating strategies, but there may be others. So again, like talking about arms races. So if you disrupt signals so that swarm mating is not ideal anymore, do you think that there's a chance that then the mosquitoes change their mating strategy and the one that's now low will become frequent, and then your strategy doesn't work anymore because they adapt mating behaviors?
I mean, yeah, certainly, yeah, that could be an adaptation, but I think that it would still have a strong effect. So I don't think that, like, for example, insecticides, there are behavioral adaptations to insecticides, and that's the challenge, because obviously you are killing less mosquitoes that you would ideally like to kill. But I think if that happens, we are in a good, you know, we are still able, we would be already able to develop a very impactful tool, you know, and that would be, because then what you ideally want is to combine different strategies, and then, so this is not gonna substitute any, it would be just a second, for example, one problem, as I was saying, is outdoor biting mosquitoes. So mosquitoes that bite outdoors, so they are not reached by these tools. But so if you are able to contribute, and then those outdoor biting mosquitoes will still mate in swarms. So if you are able to say, okay, we are targeting these outdoor biting mosquitoes by these tools, then you might be able to contribute and create targeted the broader population of mosquitoes. But yes, an adaptation, yeah, could happen. But again, I think it would be a good problem if we get there.
Yeah, thank you.
Just one comment in relation to what your question. So the objective is not to eradicate mosquitoes. The objective is to disrupt pathogen transmission. So if you can produce a reduction, even if it's seasonal or temporal, you can succeed in the eradication. We have the example of malaria in Europe. The transmission was disrupted, and now we still have the vectors, but we don't have longer transmission, and even the malaria, the Plasmodium falciparum, that is now circulating in Africa, is not able to reproduce in the mosquitoes, in the European mosquitoes. So this disruption was enough to finish the problem. So we must not think on eradication of mosquitoes, because we will eradicate ourselves before. So.
So I'm a complete ignorant on this topic, but disruptive mating system, that is acting on adults. Is that the most efficient? It seems that acting on larvae that are in water may be much more efficient. How would you apply this in open habitats? How would that go?
So yeah, I mean, larvicides are used, and it's a method that is used to control mosquitoes. So, so there are already some tools that are used to, that are attractive sugar baits, so they are used outdoors. They are used to deploy insecticide, that they are mostly trying to target these outdoor biting mosquitoes. So they consist of let's say, some feeding stations, so that mosquitoes are attracted to, that they try to mimic the scenes, the composition of the sugar. So male and female mosquitoes feed on sugar, then females will also feed on sugar, but they all need like a nectar or sugar source to survive. So they try to mimic those sugar sources, and then they deploy insecticides to them. So you could, in principle, for example, use those to deploy the compound. There are also other, there's quite a lot of effort being done to target these outdoor biting mosquitoes. So for example, there are these spatial emanators, which are basically stations that will deploy an insecticide or a repellent. So you could, if you find a molecule that is volatile, you could also employ one of those to deploy the compound, the mating disruptor. But yeah, but again, we need to have the first debated disruptor and proven in the lab, showing in the lab by feeding on so that we see an effect on mating, and then we could conduct all these field studies to see if we really see an effect on the entomological parameters, let's say.
No more questions? Okay, so
Thank
that
you for
was
your
it.
attention.
Thank you very much for coming. See you next week for the last seminar of the year before summer.